Today we are here to listen to Professor Arun Shukla. He is a professor in the Department of Bioscience and Bioengineering in IIT Kanpur. So, as I told you in the student interaction session that I don't have to formally introduce Arun because he himself is so popular in Indian academicians and science that you should already know about him. But if I am allowed to give you an example of how much we know about Arun, I told in my lab that all of you should go to attend the interaction and seminar of Arun. Then one of my students, he said, "कौन अरुण ना? वो टीपी में काम करने वाला?" So we have already circulated so many emails about this seminar, and it is a special privilege seminar series. Like we have named it as Distinguished Seminar Series. So we should be more prepared to know about the guest. Now, if you ask me about Arun, so I can give you one-line introduction that ever since I know Arun, I compare him with Roger Kornberg. If you people know about Kornberg, he is the father of transcription, and he comes from a background of academicians and he himself is a Nobel laureate. So Arun, although he has not yet got the Nobel Prize, but he comes from a background of academicians. And at a national level, he has got the fellowship of all three academies. He has got the Shanti Swarup Bhatnagar Award, Wellcome Trust Senior Fellowship, National Bioscience Award, B.M. Birla Science Award, Young Scientist Platinum Jubilee Award of National Academy of Sciences India, CDRI Award, and Sakuntala Amirchand Prize of ICMR. In addition to that, he has also got the EMBO Investigator Award. So eventually, I wish that he should get a Nobel Prize someday and stay up to my expectation. Now, coming to the work of Arun, he is a basic scientist with interest in the area of GPCRs, G protein coupled receptors. He will be talking about that in detail, but if I can just broadly tell you, because in Translational Health Science and Technology Institute, we understand things in terms of drugs and vaccines. So few of the very popular drugs, if I can name, like metoprolol or telmisartan, which are used in treatment of blood pressure or coronary artery disease, those are the drugs that target GPCRs. So there are a lot of such drugs which target GPCR, and so his work is relevant not only for understanding the cellular signaling pathway, but also for development of drug and for improvement of human health in a broader way. So with that much introduction, I will now invite Arun to please tell us about his work.
Yes. Thank you very much, Milan, for embarrassing me. I'll ask you afterwards who was that student. But it's only fair that I return the favor a little bit. So before I start, let me tell you a small story about why and how I'm here. So I'm here because of 236 rupees. So it was July 2017. I met Milan in a meeting in Mumbai, Lady Tata Memorial Trust, and we chatted for quite a while. And once the meeting was over, then I had to catch a cab to go to the airport. And for some reason that time, either the Ola app was not functional or my cell phone had problems with the 3G network. So Milan basically called the cab for me using his Ola app on his phone. And I think he had his Ola Money account set up. So he knowingly or unknowingly or unwillingly paid for my Ola ride. So that was rupees 236. And I kept telling him, and after that, 2017, we are meeting now after 6-7 years. So I kept telling him that one day I'll come and return that 236 rupees. I'm still to do that. I have not done it yet. But when Milan called me for an online seminar last year, I said, no online, I'll come in person to return back that 236 and maybe talk about science as well. So here I am. And before I start, it just so happens sometimes, we are so engrossed in science and research, we lose track of many important things. So I had an invitation from Milan, one invitation from RCB. I knew that these two institutions are close by. I did not know that they're so close by, like within the same campus. So I said yes to both of them. I thought I'll club the trip and all that. So I landed up here Monday morning, gave a talk at RCB, that is for RCB, but in the same auditorium, same seminar room. And now I'm again here for giving a talk to you guys for THSTI. So if any of you were there in my talk on Monday, I must warn you, Prasenjit was there. It's going to be the exact replay. So please pardon me about that. But sometimes replays are good, as you know. All right. So the research program in our lab is focused primarily on understanding structure, function, and regulation of G protein coupled receptors or GPCRs. I'm sure that most of you are familiar with these receptors. You must have studied about them in your biochemistry or cell biology textbook. So these receptors are integral membrane proteins present at the cell surface, and they're also known as 7 transmembrane receptors because they span the membrane 7 times. That is, there are 7 transmembrane helices. There are N-terminal acids outside the cell, there are C-terminal acids inside the cell, 3 extracellular loops and 3 intracellular loops. All GPCRs have the same conserved architecture. And these receptors constitute the largest family of cell surface receptors in our body, about 1,000 members, and they recognize a diverse set of stimuli. It can be photon of light, lipid, protein, peptides, small molecules, hormones, and pretty much nothing happens in our body without a GPCR being involved. So for example, the way you are able to look at the slide or look at me, when a photon of light hits the retina, there is a GPCR called rhodopsin that becomes active and a signaling cascade sets in, and therefore you can see things. The way you can tell about the perfume of the person sitting next to you because you have these olfactory receptors in your nasal epithelium that allow you to sort of sense the smell. The same way where you can tell, the coffee is bitter or sweet because you have taste receptors in your tongue epithelium, which are GPCRs that allow you to differentiate between different kind of taste. The way your heart is beating, the way your blood pressure is regulated, the way your brain works, the way your behavior is determined, your immune response, the way you feel hungry, the way you feel full. All these mechanisms, they have one or the other receptors involved. And therefore, it is not surprising that about 1/3 of the currently prescribed medicines, they target GPCRs. Hypertension drugs are one example, drugs for heart failure, drugs for mental disorders are some of the other examples. Even drugs for allergy that some of you might have taken. And therefore, it is very important to understand how these receptors work. That is, how they are activated, how they recognize the ligand, how they signal, and how they are regulated in the body. Now, there has been a lot of work in this area, and based on that, we have a paradigm of GPCR signaling and regulation. This is a simplified schematic, and I'll take you through this. So when an agonist binds to a GPCR, agonist is an activating ligand for a receptor. The receptor undergoes a conformational change, and that leads to coupling or interaction of heterotrimeric G proteins that is composed of alpha, beta, and gamma subunits, hence the name G protein coupled receptors. Subsequently, heterotrimer G protein dissociates, the heterotrimeric assembly dissociates. These G proteins become active. You have second messengers such as cyclic AMP, inositol phosphate, calcium, and downstream signaling. And this is what we refer to as classical G protein dependent signaling. Now, this has to be tightly regulated Otherwise, the cellular homeostasis will be a complete mess. So for that, the cells have designed or devised a mechanism that is known as desensitization. And essentially what happens is that activated receptors are phosphorylated by a family of kinases known as GRKs, GPCR kinases, which phosphorylate the receptor either in its C-terminus or sometimes in the loops, intracellular loops, because that is inside the cell. And that phosphorylation leads to binding of a scaffolding protein, multifunctional protein, known as beta-arrestins, and you'll be hearing more about it today. And it was initially believed that binding of beta-arrestins essentially occludes the binding site for G proteins, and hence, once beta-arrestins are bound, G protein interaction is terminated, or G protein signaling stops. But we have found over the last 2 decades or so that in addition to stopping G protein signaling, beta-arrestins have multiple other functions. For example, they can also mediate receptor endocytosis, that is, trafficking of the receptor from the plasma membrane to the endosomal vesicles. And beta-arrestins can do that by working as a scaffold for clathrin, adaptin, and so on. They can also bring E3 ubiquitin ligases close to the receptor, leading to receptor ubiquitination, degradation, and so on. But most importantly, it turns out that beta-arrestins have their own signaling capacity, which is independent of G proteins, meaning they also initiate a parallel wave of signaling that is in parallel to G proteins. And before I proceed further, let me tell you that I've tried to keep things simple. I will not go into too much of the details, but feel free to stop me anytime and ask questions. And we will be talking more about this part of the paradigm today. So unlike what is there in the textbook, essentially GPCR signaling is bifurcated. It can happen through G proteins, that is the classical mechanism. It can also happen through beta-arrestins. And the reason why this is interesting is that these 2 signaling pathways often lead to very distinct cellular and physiological outcomes. And this has very, very interesting implications, because this has given rise to what we refer to as the framework of biased agonism. So what is biased agonism? That is described here in this slide, and I'll just quickly try to explain this to you. So antagonist is a ligand that essentially stabilizes an inactive conformation of the receptor. That is, there is no G protein, no beta-arrestin signal. So receptor is silent. When an agonist binds, that we also refer to as unbiased ligand, this can be the natural ligand for the receptor, hormone, for example. Then you have both arms of signaling, G proteins and beta-arrestins. But it is possible to design ligands which will selectively engage either G protein or beta-arrestins. For example, you can design a ligand that will bind to a receptor, activate only beta-arrestin pathway, but not G protein pathway. And such a ligand is known as G protein or beta-arrestin biased ligand because it biases the signaling towards beta-arrestin. Right? Similarly, you can have a G protein bias ligand, which will bind to the receptor, activate G protein signaling, but not beta-arrestin signaling, and that is known as a G protein bias ligand. And these are synthetic ligands that we can design and develop in the lab. And this has major implications for novel drug discovery, and I'll try to explain that to you by giving you one example. So morphine, I'm pretty sure that most of you must have heard about. It's a very powerful analgesic. So this is given to the patients after for acute pain, after surgery, for example. This is very potent analgesic, but it also has side effects, constipation, respiratory distress, and so on. It turns out that it works by activating a GPCR known as mu opioid receptor. And it's a full agonist or unbiased ligand for mu opioid receptor. When it binds to the receptor, it activates both G proteins and beta-arrestin pathways. It turns out that the G protein pathway is responsible for pain relief or analgesic effect. The parallel beta-arrestin pathway gives rise to constipation, respiratory distress, tolerance, and so on. So now if you conceive a ligand, which is a G protein biased ligand, it could be a modified version of morphine or a new pharmacophore altogether, what you have is only G protein signaling because the ligand is G protein biased, and then you have analgesia, but you don't have the side effect, right? So using the same target, now you can get rid of or minimize the side effects. And this used to be a conceptual framework until a couple of years ago. In 2020, FDA approved the first in-class biased ligand named as oliceridine, which is basically a G protein biased ligand at mu opioid receptor as an alternative treatment for acute pain in place of morphine. And this is superior because it reduces the side effects without having any effect on analgesic effect of the ligand. Now, this is one example. There are several examples. In fact, most of the GPCRs which I've studied in this context, it seems that sometimes it's the G proteins which are beneficial, sometimes beta-arrestins are beneficial, and therefore this has opened up the new possibility of novel drug design in the industrial context, for example, because you can now design better drugs at the same target by simply playing with the signaling profile or functional selectivity of the ligands. And this has changed this landscape of drug discovery in major pharma. Now, every major pharma has a program along these lines for developing biased ligands, because you don't have to discover new targets. Using the existing targets, you can, pack or package the drugs as a new drug.
What bifurcation will be followed?
Very good, excellent question. So at this point, we believe that the ligand structure or ligand efficacy will have a specific conformational correlation with the receptor, meaning a G protein biased ligand will stabilize a conformation in the receptor that will allow G protein coupling but not beta-arrestin coupling. So it's the receptor conformation induced by ligand binding that determines the subsequent transducer coupling. Okay. All right. Now, these ligands that I described, giving one example, these are all synthetic compounds, mostly developed and designed in the lab. But as it so happens, many of the things that we discover in the lab, nature has already discovered it or designed it in one form or the other. And therefore, we ask the question, if we can bias these receptor signaling by designing chemical entities, is it possible that nature has already designed biased receptors? We just didn't know about them, right? And if you go through the literature, it turns out that there are 7 transmembrane receptors that were classified as non-signaling or non-functional or non-canonical GPCRs. Why GPCRs? Because they have 7 transmembrane architecture. But why non-signaling? Because they do not couple to G proteins. And earlier, the only known signaling mechanism was thought to be G proteins, right? Now, it doesn't make sense that nature would design something which is non-functional. It's going to take space in the membrane. It is going to be energetically quite expensive. So it doesn't make sense that you will design something. Nature will design something without any function. So we asked the question, is it possible that these receptors may signal through beta-arrestins, right? So first we looked at interaction of beta-arrestins. And there are several examples, complement receptor, and these are all chemokine receptors. And we're using one of the chemokine receptors, DQI-D6 receptor, as a model system. And this is a very simple experiment where we co-express the receptor with beta-arrestins, and we do a co-immunoprecipitation assay where we IP, that is, pull down the receptor using a tag. And basically by Western blot, we look for co-purification or interaction of beta-arrestins. And it turns out that indeed, this so-called non-functional GPCR, it does interact with beta-arrestins in response to agonist. This is a chemokine agonist. There are 2 isoforms of beta-arrestins, beta-arrestin 1 and beta-arrestin 2. And this receptor D6R interacts very specifically to both the beta-arrestin isoforms. We can also validate it using confocal microscopy, where we co-express the receptor with GFP tagged beta-arrestin. And in response to agonist stimulation, the receptor goes to these endosomal vesicles where it is co-localized with beta-arrestins, meaning not only they recruit or bind beta-arrestins, but they can also have endocytotic function in this context. That is, beta-arrestins are promoting endocytosis of the receptor. So these so-called non-signaling or non-functional receptors are indeed arrestin-coupled receptors. They interact with arrestins or beta-arrestins. What about signaling? Now, the canonical GPCR signaling we tested, but they were not operational downstream of these receptors. So we did global phosphoproteomics study using mass spec-based approach or phosphoantibody array approach, where essentially the idea is to test the phosphorylation, dephosphorylation of all the cellular proteins in the cell expressing these arrestin-coupled receptors and see which proteins undergo phosphorylation, dephosphorylation as a proxy or a readout of potential signaling pathways. And I will not go into the technical details, but we found a whole lot of proteins which were showing an increase in phosphorylation or decrease in phosphorylation in response to agenous stimulation for cells expressing the arrestin-coupled receptor. And these proteins can be classified based on biological roles, cellular localization, and molecular function. And we have subsequently shown that these potentially signaling networks are indeed operational downstream of D6R by using beta-arrestin knockdown, inhibitor-based approach, and so on. So these receptors are not only arrestin-coupled, but these are arrestin signaling receptors. That is, they are signaling through beta-arrestins. All right. So essentially, unlike GPCRs, which couple to G proteins and beta-arrestins both, these arrestin-coupled receptors, or ACRs, lack G protein coupling, but they do couple to beta-arrestins and signal through beta-arrestins. I gave you an example of D6 receptor, which is one of the members of this subfamily. Now we have discovered almost a dozen different arrestin-coupled receptors, and this all seem to follow this pattern. So this is a new subfamily in the large subfamily of GPCRs that are indeed arrestin-coupled receptors, which are naturally present. And these are the natural examples of beta-arrestin bias 70MRs because we are not stimulating them by any synthetic ligand. We are stimulating them by their endogenous ligand, naturally occurring ligand, and that ligand is able to induce only biased signaling. Okay. Now, one of the things that we are interested in going forward is to visualize all this at structural level to see what are the structural determinants that govern the transducer coupling. If a receptor will couple to both G proteins and beta-arrestins, or only to G proteins, only to beta-arrestins.
So Arun, just clarify, are they promiscuous? I mean, are they dedicated arrestins or
yes.
pathways? How do we...
Right. So we will. I will come to that. What we have found is that there are only two isoforms of beta-arrestins, of course, and the paradigms that I have talked about in terms of endocytosis, signaling, ubiquitination, that is broadly conserved, meaning these two beta-arrestins are able to interact with and govern all these receptors. And how that happens, that I'll come to in the second part. All right. So we are taking complement receptors where there are three different 7 transmembrane receptors in the complement cascade. So complement cascade, as some of you might know, it's a part of our innate immune system. And these two complement peptides, C3A and C5A, these are very potent anaphylotoxin, which are generated upon complement activation that can happen through virus or bacterial infection, upon viral or bacterial infection. And C3A binds to and activates C3AR, one 7 transmembrane receptor. C5A binds and activates two different complement receptors known as C5AR1 and C5AR2. Now, C3AR is a prototypical GPCR where it activates G proteins as well as beta-arrestins. But C5A, as I said, it binds to C5AR1 and C5AR2. C5AR1 is a prototypical GPCR, activates both G protein and beta-arrestin, but C5AR2, despite binding the same complement peptide, that is C5A, is a naturally biased receptor, that is, it couples to and activates only beta-arrestins. And now we have determined a whole series of structures of these receptors bound to G proteins, bound to arrestins, and that has allowed us to visualize what determines their selective coupling to G proteins or beta arrestins. But that is not for today, maybe next time when Miral calls me. So I'll come back to beta arrestins. Is the air conditioning not on, or is it just me who's feeling hot? Now, beta arrestins, as I mentioned, there are only two isoforms, that is beta arrestin 1 and beta arrestin 2. And sequence-wise, they are about sequence identical. And if you look at their structure, basically they have two domains, N domain and C domain. But the paradigms of endocytosis, ubiquitination, signaling that I mentioned, they are conserved across all the GPCRs. What it means is that these two isoforms of beta arrestins are able to somehow interact with and regulate this large repertoire of GPCRs. And that begs the question, is there a universally conserved mechanism for GPCR beta arrestin interaction? And this is an interesting question because if you look at the primary sequence of GPCRs, it's not very well conserved. It means that it cannot be a sequence-based recognition of GPCRs by beta arrestins. It must be some other mechanism. And that remains a frontier question in the field, and that is where we have tried to do a little bit, which I will share with you. Now, in terms of GPCR beta arrestin interaction, it's a biphasic process. So GPCRs are activated, then they are phosphorylated by GRKs, GPCR kinases, in C-terminus or in the loop, and then beta arrestins will come and bind. And in the fourth step, they will interact primarily through the phosphorylated tail, and that is known as tail engaged or partially engaged complex. That is step 1. And then in the second step, beta arrestins will further interact with the transmembrane bundle or transmembrane core of the receptor, making what we refer to as fully engaged or core engaged complex. Now, one thing that is common in all GPCRs is that they all undergo phosphorylation upon activation. So we thought maybe the clue for universal interaction between GPCRs and beta arrestins could be somewhere here. And this is what we have focused on in the past year or so. Now we have taken vasopressin receptor as an example of a GPCR. It has multiple phosphorylation sites in its carboxyl terminus or tail. And we have made a whole combination of these mutants where we have changed these individual sites to alanine, either one by one or in different combinations. And then we have essentially looked at how does individual or combination of mutants, how do they affect beta arrestin recruitment, trafficking, signaling, and so on. And I will not go into the details as I promised, but what we have found by doing all these assays, that despite having these multiple phosphosites, it's a small pattern that you can read as PXPP that is most crucial for beta arrestin interaction and activation. And P here is a phosphorylation site, and X is any other amino acid. In case of vasopressin receptor, it's alanine. Now, even in this PXPP, it turns out that at least in V2R, vasopressin receptor, this particular site, 360, is most important. And what I mean by that is if we make a single phosphosite mutation, that also significantly influences beta arrestin interaction and trafficking. So this is a simple confocal microscopy experiment where we are comparing the wild type receptor with a single phosphosite mutant of the receptor referred to as T362A. What we are looking at is GFP tagged beta arrestin 1 in this case. So in the basal condition, beta arrestin is localized in the cytoplasm. In response to agonist, at early time point, it goes to the membrane, and that is where it interacts with the receptor. At late time points, beta arrestins and receptors, they basically get internalized in these endosomal vesicles. That is typical wild type pattern. In case of the single phosphosite mutation, beta arrestins in the cytoplasm under basal condition, they go to the membrane. But even after prolonged exposure, that is late time points, they stay at the membrane, they do not get internalized. So a single site is critical for trafficking of beta-arrestins. And similarly, we have found, individual sites could also play a major role for other receptors. But this is a population type study where we are looking at a cell population, multiple cells, and a pool of beta-arrestins or receptors. And then we have moved on to single molecule level, not high resolution, but essentially trying to tag the receptor and beta-arrestins differently using different fluorophores, and then track them, their movement, trafficking in live cells using single molecule tracking. And I will not go into the details of this study, but what we have found is that unlike what was generally believed in the field, that is receptor beta-arrestin stable interaction is the key for beta-arrestin activation and signaling. What we have found is that beta-arrestins, once they're primed by the interaction with receptor, then the receptor can fall apart and beta-arrestins can tether themselves into the membrane, that is the plasma membrane, and continue to maintain an active conformation and carry out downstream signaling. How exactly that happens? Now, we would like to understand that at high resolution. This is single molecule tracking where each of these dots are indicating one individual receptor, but still it's done in the live cell. We are, at low resolution, we are not looking at atomic details. Now, we are moving to the level where we can really try to visualize all this at high resolution, hopefully in atomic details or near atomic details. And that is where we use cryo-electron microscopy as one of the approaches. The idea is that we reconstitute these GPCR beta-arrestin complexes and visualize them by cryo-EM to get a three-dimensional structure and see what is the universal mechanism for their interaction and beta-arrestin activation. So for this, we are taking beta-1 adrenergic receptor as one of the receptor examples. We purify it, we put it in nanodisk. Nanodisk is a lipid-like environment where the receptor feels at home because the receptor is a membrane protein. So we purify and then we put it back in the lipid-like environment. Then we add purified beta-arrestin, stabilizing antibody fragments, and then we reconstitute this complex and then get the structure by using cryo-EM. And in this case, you see beta-1 adrenergic receptor here in white. This is the ligand or the agonist that was used. It's known as formoterol. It's a long-acting beta agonist. This is the ligand that is present as a drug in asthma inhalers many times. And then in yellow, you see beta-arrestin. And then this is the stabilizing antibody fragment that helps lock this complex together. Now, if we zoom in a little bit, this is beta-arrestin. And this in green, you see the carboxyl tail of the receptor that has multiple phosphorylation sites. So it docks in the end domain of beta-arrestin. Why end domain? Because end domain has this electropositive groove, and that is where these negatively charged phosphates attached to the receptor C-terminus like to go and sit. If we zoom in a little further, we see that this particular tail has multiple phosphates, but once again, three phosphates which are aligned in PXPP or organized in PXPP pattern, they make most of the contacts in this binding interface. These three phosphates, 360, 362, 363, they interact with multiple lysines and arginines which are present in the end domain of beta-arrestin. Now, this is one receptor, and what we are looking for is a universal mechanism, if there is any. So now we have established a pipeline where we have determined the structures of almost a dozen different GPCR beta-arrestin complexes. And we have set up the protocols in such a way that if we collect data in the morning, cryo-EM data, we will have a structure before dinner. And we have done almost a dozen different structures, as I said, and I'm just going to show a couple of them. So this is CXCR4, a chemokine receptor. This is a complement C5AR1 receptor, and they're both complex with beta-arrestin 1 in this case, and we have the structure. Then we have the other isoform that is beta-arrestin 2 in complex with vasopressin receptor or D6 receptor that is an ACR, arrestin coupled receptor. And of course, they all look the same. These are similar beta-arrestin molecules. And the C-tail of the receptor is shown here in this beta strand. What is important here is that these receptors, they have different primary sequence. They have different organization of phosphorylation sites, and they have different number of phosphates in their carboxyl tail. Okay. So we take all these dozen different structures and then we superimpose on top of each other. And it turns out that all these receptors, their phosphorylated tail, it docks to the same end domain electropositive surface, right? If we zoom in further, what we see is that the three phosphates from these different GPCRs, three phosphates which are aligned in the PXPP pattern, they cluster together. 1, 2, 3. The other phosphates present on these different receptors, they're resolved in the structures, but they're more scattered here or here.
Do you have ligand also in these complexes?
So in these complexes, we are using the phosphorylated receptor tail only, not the entire receptor. So there is no ligand per se. Now, if we look at it more closely, what we see is that, as I showed earlier, in case of beta 1, again, these three phosphates make a very strong interaction with lysines, arginines, which is organized in a KRK type motif. And these different receptors, they have different phosphorylation pattern, different primary sequence, but they sit on beta-arrestin in a way that PXPP pattern that is present always makes the same contacts. So this is potentially a universal mechanism that allows these different receptors to interact with only two isoforms of beta-arrestins. Now, this is all done structurally. Can we really confirm it in cellular context? Obviously, we cannot determine the structure of all 800 receptors. So we have done sequence alignment of the entire GPCRome, that is all 826 receptors. And we find that most of the receptors, they have this PXPP motif in their carboxyl terminus or in their third intracellular loops. So this motif is present in a majority of GPCRs and ACRs. Now, to test this in the live cell context, in cellular system, we use an antibody-based sensor to monitor beta-arrestin activation. And this is a very simple assay. So we co-express receptor, beta-arrestin, and an intrabody sensor. It's called intrabody 30. So this intrabody is basically a cytoplasmic version of an antibody fragment, a synthetic antibody fragment, that recognizes beta-arrestins only in its active conformation. What I mean by active is when beta-arrestins are bound to the GPCR. So it is a sensor for activation of beta-arrestin. Receptor is expressed, intrabody is fused to a small fragment of nanobit, that is a luciferase enzyme. Beta-arrestin is fused to the other fragment of the luciferase enzyme. The idea is that when beta-arrestins interact with the receptor, the intrabody will go and recognize receptor-bound beta-arrestin. Hopefully, these two fragments of the enzyme will come together. There will be a complementation. If you add the substrate, you'll measure the luminescence. So your luminescence signal is basically a readout of beta-arrestin interaction and its activation. Very simple. Now we look at a couple of different receptors. Chemokine receptor CXCR3. It has PXPP in its C-terminus. In response to agonist, we see very nice dose-response curve indicating beta-arrestin interaction and activation. We look at muscarinic receptor, M2R. It has PXPP in ICL3. Once again, in response to agonist, we see this nice dose response, beta-arrestin interaction, and activation. In case of CXCR7, another chemokine receptor that lacks PXPP, and we basically see a flat line. That is, beta-arrestins are not getting activated, at least in a conformation that can be read by this intrabody sensor. These are natural receptors, of course, wild-type receptors. We have also done gain of function and loss of function experiments. So this is complement receptor C5AR1. It has a PXPP, and as you can see here in black, in response to agonist, very nice activation of beta-arrestin. If we do site-directed mutagenesis to break this PXPP, disrupt the PXPP, by adding one more amino acid here, we lose beta-arrestin activation to a significant level in blue. Bradykinin receptor, it does not have a PXPP in its C terminus. In black line, in response to agonist, we basically get a flat line. There is no beta-arrestin activation as recognized by this intra-body sensor. If we reconstitute PXPP by site-directed mutagenesis, we get very nice activation of beta-arrestins. So essentially what it tells us is that there is a lock and key type mechanism that is operative here, where the PXPP serves as a key, and PXPP being present in GPCRs or ACRs. And this essentially engages this KRK lock that is present in beta-arrestins. So in the basal state, these lysines and arginines are stabilized through intramolecular interactions. There are certain glutamates and aspartates in the C terminus of beta-arrestin that is docked in the end domain when the receptor is not there. And this sort of engages the charge of these lysines and arginines. So beta-arrestins are in a locked state, or the brakes are on, if you will. When a receptor binds, then basically the C-tail of the receptor that is phosphorylated removes or pushes away the C-tail of beta-arrestin, and the receptor-bound phosphates now engage these lysines and arginines in the end domain of beta-arrestins and thereby activate beta-arrestins. Yes. Yes. Yes.
Can you please explain the x-axis?
So basically, so this is the concentration of ligand, molarity in log. So -6 means 1 micromolar, and 10 micromolar, 100 nanomolar, and so on. Okay. So what it means is that as we increase the dose of the ligand, then only we are getting an increasing response, which means it's a specific response, because by increasing the ligand concentration, we are activating more and more receptor. Right. So this PXPP key opens the KRK lock in beta-arrestins. So basically what I have told you is these two main things, key things. That is the discovery of natural examples of beta-arrestin bias 7 transmembrane receptors that we refer to as ACRs, or arrestin-coupled receptors, and a conserved lock and key type mechanism of GPCR phosphorylation-dependent beta-arrestin activation. These are the people who do all the hard work. Most motivated, sincere, and hardworking students that I know of. They are the real superstars in the lab. I basically sit in the office and take credit for all their hard work. I consider myself very blessed to have such a wonderful set of students who inspire me every day. We have a very extensive set of international collaborations, because no matter how smart a person is, you cannot become an expert in everything. And in biology, it's very important to address a question in a holistic manner. So we basically try to bring together different expertise, complementary expertise together on a table so that we can answer a major question using multiple orthogonal approaches. And it has been a very enjoyable feeling and experience to work with all these leaders in the field. And we continue to stay in touch, discuss science, design projects, troubleshoot through Zoom meetings and Skype and so on. It has been a very pleasant experience so far. We are supported by SERB, DBT, Welcome Trust India Alliance, IT Kanpur, EMBO, DBT, DST, ICMR. And we are very thankful to these funding agencies for trusting our research program and putting their faith in us. When I moved back to India back in 2014, April 2014, at that time, there were 0 cryo-EM facilities, high-end facilities in the country. And we have been working with the funding agencies, policy-making bodies behind the scenes. And now there are almost 10 high-end facilities in India all across the country, supported by DBT, SERB, CSIR, including one that we are setting up at IT Kanpur. This is the next generation cryo-EM. Even Columbia or Yale does not have this setup yet. This is most sort of advanced setup And all these facilities are national facilities, which means if one day you feel that you need access to cryo-EM, you can go and use these facilities. So in addition to doing science, working with these funding agencies and sort of having contributed to this effort of establishing these high-end centers has been very satisfying. And obviously, this is not an individual effort. Many, many people, many scientists in India, they have worked together to make this a possibility. This is certainly not an individual sort of effort. Now, I'm done with the science part. And with Chair's permission, I would like to share a couple of anecdotes of my scientific journey in Indian science. All right. I'll take only 7-8 minutes, not more than that. So it was, as I said, I moved back in April 2014, and I was presenting my vision for the research program in front of a committee in July 2014, 3-4 months after I had returned back to India. And my vision, meaning basically what I described today, I was saying that this is what I plan to do in India as an independent investigator. And it was not a committee at IIT Kanpur, it was somewhere else. The committee had 13 people, and one of the committee members who happens to be, and this is again for the students, the faculty colleagues can completely ignore it. They might feel that bahut gyan baant raha hai, so that is, you don't have to pay attention to it. It's mainly for the students. So one of the committee members, who is one of the most decorated scientists in India, this award, that award, director here, director there, he looked at me and said, and I quote, "Come on, who are you kidding? This cannot be done in India." So I looked at the other committee members. A couple of them were dozing off in their seats. I don't blame them, but even people who were awake, nobody uttered a word. So then I looked at this committee member and said, "Thank you very much for your feedback. We are still going to try." And we did try. Whether we have been able to contribute a little bit or not, I leave it up to you to judge. But that is not the end of the story, because if it was, I will not be sharing this with you. It turns out that the same person over the years has become one of our biggest fans. That is one of the biggest fans of our research, our work. So if you believe in yourself, if you have confidence, if you have the ability to take criticism as motivation, you can even turn your biggest critiques into your allies. And that is something for you to think about. Now, that happened 3-4 months after I moved back to India. I'm not saying these things in a negative light. This is important for the students to sort of see if they can get some message, some motivation. And that is why I like to share these things. Now, when a new faculty joins, for example, at many institutions, more experienced people like to share their wisdom. And I was also called by someone to share that wisdom. And the person said, very experienced researcher, that if you have foreign collaborators or foreign authors on your research papers from here, this will not count for your promotion or any recognition or your career growth. And I was, basically shocked, because all my PhD and postdoc, what I had learned is that collaboration is key to do cutting edge and frontier science. Science is a collaborative endeavor. And I basically, heard it from here, let it go from the other ear. I kept hearing it from one place or the other over and over. People will even write that in email. Not only oral, but even go on record to say that. I kept ignoring it. But then one of the fellows from the lab was interviewing for a faculty position. So PhD in India, postdoc with us. And he was told the same thing by the selection committee. And this member was not the same as the earlier person, but a different person. But again, a very decorated scientist, all the awards, all the directorship, whatnot. And he said to this fellow who was interviewing that, your papers have foreign authors. I'm not sure how you are even here, how you are even called for the interview. You should not be called. So that fellow came to me and explained. And that is when I decided to write this article. So what I'm trying to tell you is that no matter what people tell, what you hear, science is collaborative endeavor. Do collaborate. Do not hesitate at all. And if you collaborate, your experience is going to be very good. Science, you can really address questions in a holistic manner. You can contribute in the frontier areas in cutting edge problems through collaboration. This is my last slide, by the way, so I'm not going to take too much of your time. But one of my sort of biggest joys of being in Indian science in the past 9 years has been our students and fellows. As I said, I consider myself very blessed to have wonderful students in the lab. I've seen students abroad, in Europe, in States, and I was mentioning it to those students who were there in the interaction session earlier. And students, many of them, they mind their own business and then they'll come do their work and go home. Here in Indian sort of context, I have seen that people have much more collegiality, team spirit. किसी का काम नहीं कर रहा है कोई experiment. So ऐसे नहीं कि मतलब नहीं दूसरा उसको sort of, they'll come give a hug and say, चल चाय पीने चलते हैं, चाय पी के आएंगे फिर देखेंगे क्या होगा. That kind of team spirit, that kind of collegiality, I find it very touching. It means that you have a supporting network of students and colleagues in the lab. And that is very, very important. And I believe that you guys are the smartest people. That is the students. Faculty are also smart. But you have what it takes to do really, really well in research. Sky's the limit. And I really wish, and this is one of my dreams, that one day one of you goes to Stockholm to get a Nobel Prize. And when you do, don't forget me. Thank you very much. So I'll be happy to take questions. Yes.
Thank you, Arun, for this very excellent presentation, and especially for the last part for motivating students and maybe all of us, including everybody. Now the talk is open for questions or any discussion. Feel free to ask.
Yes. Thank you.
Any situation is like young arts internal can.
That is a very good question. So for GPCRs, as far as I know, there is no known instances for them flipping. The only time it will come close is if you look at their orientation in the plasma membrane versus endosomal vesicles. So when endocytosis happens, when plasma membrane pinches off, that is when the N-terminus will go inside the lumen of the endosome and the C-terminus will be outside like that. But that is still happening inside the cell. So as far as I can recall, I'm not aware of any instance where N and C-terminus, they are flipped.
How this can happen when there is no chemical involved, like receptors?
Yes. So mechanosensation is a fascinating field. And again, the entire mechanism is not clear, but many of these receptors, they have some basal level of activity that we refer to as constitutive activity. And what it means is that even in the absence of ligand, these receptors are not rigid. They are not brick-like structures. They're very flexible and dynamic. So they are sampling all different conformations, and maybe by chance, if they hit an active-like conformation, that is enough to give you some basal signaling, because G protein is always close to the membrane. Receptor adopts that active-like conformation by chance, and that can give rise to some basal signaling. In case of mechanosensation, it may be the sort of stretch of the membrane where the receptors are present that can allow the conformational change in the receptor and thereby activate it. Yes.
Yes.
Yes, yes.
Any specific database or people can find well-derived metabolites with the potential and use for contamination or degradation.
This is a fantastic question. So for metabolites, I'm not sure that such a database exists. There was a preprint where Dr. Gaurav Ahuja from IIIT Delhi, he is trying to look into something like that in terms of G protein activation by using sequence analysis and trying to figure out if some metabolites can act as agonist antagonist. But as such, as far as I'm aware, there is no well-curated database, but that could be very, very interesting if we have one. There are receptors like free fatty acid receptors, for example, which do use metabolites as their agonists or antagonists. Yes, that'll be fascinating if we can have one. Yes. Yes.
What is arresting named because they arrest the GPCR binding with the G proteins?
Yes, so that is how they were named originally, because they were discovered to basically terminate or arrest G protein signaling.
Yeah, so now you have shown a new paradigm.
Yes.
There's a parallel signaling because of arresting. So do you think that you would like to advocate for renaming them?
Right. That is a very good idea. I mean, they're still doing arrestin function. So it's not that they're not arresting anymore, but if there could be a good name which will satisfy both arresting and activating, I think that could be considered. Yes. 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 Yes.
So nice talk.
Thank you.
I was thinking of the upstream events that actually lead to the changes that we have shown.
Yes.
So when we look at ligand binding pocket,
Yes.
the regions that agonists bind are different from the regions that antagonists bind. How does that actually lead to conformational changes? That's what I'm curious about.
Right. So typically, in most cases, the orthosteric binding site for agonist and antagonist will be the same. That is, if antagonist binds tightly, then agonist cannot bind because it's the same binding site. And the idea is that when an antagonist binds, it sort of does not allow the movement of transmembrane helices so that the intracellular face of the receptor will not open up to accommodate the G protein. When an agonist binds, it induces conformation in the 7-transmembrane bundle such that there is an opening on the inside and there is a cavity formed on the intracellular surface, and that is where G proteins come and interact.
PT determines this.
Yes, yes. So if you have both agonist and antagonist, then the affinity will determine it. If not, then it's the specific interaction and distinct conformation stabilized by antagonist versus agonist. Yes.
Yes, I am actually, it was a fantastic talk. Thank you very much.
Thank you.
So, I think we spoke in the morning briefly, because primarily my work is essentially in the area of HIV.
Right.
I know there is only one FDA-approved drug, which is the Maravajok,
Yes.
which is not yet in India. I think
Right.
mostly not in the clinical government settings, which actually targets some of the receptors.
Yes.
What you just showed today is something very interesting, that how do you actually switch off one of the pathways to block what you mentioned as can cause side effects.
Yes.
Something I'm not sure has ever been done on those kind of where drug discovery. People who does a lot of drug discovery where they target protein couple receptors.
Right.
That is one area that I would like to know, not necessarily today.
Sure.
Because I have long interest in understanding how we could actually design a small molecule that can interact or interfere the interface between the virus and the chemokinase.
Yes. So that is So yes, I'll take the other question. So before I forget this one. So Mahavirok is basically a classical antagonist. So when it binds, it does not allow the receptor to get activated. Now, we have structures of CCR5 bound with G proteins and beta arrestins in complex with CCL5, that is the natural agonist. And it turns out that if you make specific mutations in CCL5, you can bias the signaling towards one side or the other side. Whether that has some implication for HIV attachment, that is not something that people have looked at. But what we are doing is we have these synthetic antibody libraries for SAFEs, FABs, nanobodies. And the idea is if we can generate antibody fragments against the external surface of CCR5, can they serve as a way to block the HIV attachment? So that is something that we're looking at. Yes.
And the other thing is, it's not a question, but something that I just saw in your acknowledgement slide. You actually mentioned a lot of Center for Excellence for Cryo-EM. And you mentioned that yours at IIT Kanpur is probably some way better than what exists. I have also seen the name of Somnath Datta, who we work closely, very closely. And I would like to know some of the facilities, because there are some of us.
Yes.
In case we want to collaborate with you.
Absolutely. Yes, yes. So all these facilities are more or less
functional capacity.
So some of the facilities are functional. That is Hyderabad, 2 facilities in Bangalore. IIT Delhi should become functional very soon. The other ones are still being set up. But all these facilities more or less at the same level. There is a slight difference here or there, but they're mostly high-end facilities, meaning you can get atomic structures. So it's not that EM setup was not there in India, but those were more for looking at large complexes, and the resolution was not going that high. But now we have the high-end setup, and more or less all the facilities are about the same. All these facilities are national facilities, and 3 or 4 which are operational, you should be able to book time and go and use it, or send samples and collect data. And it's going to be the same for SERV sponsored facilities as well. Yes. Yes.
So, nice talking. Thank you. In case where this EXPP is not present.
Yes.
And did you try to introduce them to get the activity?
Yes. So we have done in the case of bradykinin receptor just as a proof of principle, but it's not that we are doing it at a large scale, because it's like a mutant receptor, right? That is not how it happens. So we have just tried to show it that if you engineer, you can gain reactivity or activation. Yes. So in bradykinin receptor, that has happened, but we are not taking it at a large scale, so to say. Yes.
And other thing, like in case of this COVID,
yes,
people are apart from AS2. Recently, one science paper came where they are talking about THSTI is one of the reasons for the spread of that.
Right.
So in that scenario, you think that rather than targeting THSTI, if we target GRK, do you think we can get the same result?
That is a fantastic question. So in fact, yes, because phosphorylation is essential for beta-arrestin binding. And if you shut down GRKs, theoretically, you should be able to block beta-arrestins. Now, there are some instances where there could be other kinases as well, which can phosphorylate. So perhaps not in those cases, but for a large majority, yes, that will be a feasible strategy. Yes. Yes. Yes.
And last question.
Yeah.
So these 3 phosphorylation,
yes,
XXPT,
yeah,
all 3 happen together. In between that, there is no other PTM.
So we cannot say for sure whether there is any other PTM or not, because in some receptor that X can be a lysine.
Yes,
so it can have ubiquitination or something else, but that is not something that we have looked at. What we have found is that the spacing by X is critical. If we don't have X, if we have PPP, it does not work. If we have spacing too long, PXX, PP, then also it does not work. Meaning there is a certain spatial pattern that has to go and fit in the groove for it to hit all the right lysines and arginines.
Why I'm asking this, maybe if X is G,
yes,
and if X is tryptophan,
yes,
so the scenario will change because your rest 3 phosphorylations right are very close.
Right. And
tryptophan is a big.
Right.
And this agonist-antagonist movement,
right,
like in case of bile acids and others,
yes,
this tryptophan case is playing some role.
Yes. But that is a very good point, and I think it'd be very interesting to look at these sequences more closely. Yes. Thank you. Yeah. Yes, please.
In the KRK motif, did you find any of the lysine or arginine modifiers?
That's a very good question. So we have not really looked at the modifications.
Because it is known to have crosstalks of modifications.
Absolutely. You're absolutely right. So we have basically looked at it from the charge-charge interaction perspective. That is negative charge on the phosphate and positive charge on lysines and arginines, but we have not really looked at the other modifications. That is definitely something to keep in mind. Yes. Thank you. Students? Milan, yes? Yeah, you get one question for embarrassing me.
Again, I have a very naive question, so I don't want to offend anybody. So the question is, you are so experienced with CRI and you also have a lot of in silico tools like AlphaFold. So, can we really rely on those in silico predictions for doing some kind of mechanistic study? Or what is your opinion?
So we like to believe that simulation studies or computational studies combined with experimental evidence are very, very powerful. So many times when we do these structural studies, for example, then we collaborate with somebody to ask specific questions using simulation.
Before investing in the experimental agents or type.
Right.
So can we start with in silico study and then it makes sense if I have a CRI and facility and
Yes.
if I have a CRI and computer?
Absolutely. So I would say that as a hypothesis generator, as a starting point, it will be very, very powerful. Whether it will work or not, that will depend, on case-to-case basis. That will, So in some cases it can work beautifully. In other cases it may not work. But that is the problem with every approach. So if you have the facility, if you want to use that as a starting point, absolutely. What is happening, for example, now, even with AlphaFold, people are making models and doing in virtual screening to get ligands and so on. And in some cases it's working, some cases it's not. So I guess we cannot say for sure for a given target if it's going to work. But if you have the facility, if you have the resources, then absolutely it's worth a shot. But nothing is perfect, as you know, whether it is computational approach or hardcore experimental approach. Yeah, please.
Just one more question.
Yes.
Also, Punjab in insular eukaryotes, prokaryotic kingdoms, which they are all in the microscope.
So there are receptors in yeast, sterile receptor, sterile 2. In prokaryotes, there are 7 transmembrane proteins. It's a very interesting question because we have been thinking about this a lot lately. So for example, in Plasmodium, there are 7 transmembrane receptors, same architecture as GPCRs, but they do not have G proteins, right? But people have not really looked at them. But they have arrestin-like proteins. So now we are thinking from that angle, could these proteins indeed be arrestin-coupled receptors? But bona fide GPCRs, no. Something that comes close is bacteriorhodopsin, but that is more like, pump than receptor and things like that. So not really bona fide, well-established GPCRs, no.
Actually, you can go to because now there are 3 papers in the literature.
Yes. Yes.
That's very close to the
Right.
to find that similar types of structure.
Right. I think that would be very, very interesting to look into. Yes. We have not really done that, but that would be very interesting. Yes. Yeah. Thank you. All right.
Students,
It's okay if you're being shy, it's completely fine. If you have any questions later on, of course, I'll be around, or you can email me anytime and I'll try to respond as quickly as possible. Thank you.
So let's thank Professor Arun for coming all the way from Kanpur and giving this lecture. So thanks, and please don't accept that 236 rupees because that will force him to come again next time whenever we invite. And I thank all the audience as well. Prasanjita, thank you. So at the end, as a token of appreciation, I would request our Dean sir to please come on the stage and hand over the token of appreciation from THSTI.